Hey A&Pers, this is Dr. Jones checking in. We are here in the A&P1 lab room doing videos for lab 3, and this should be video number 3. So we are here with the prettiest model, my very, very favorite model. So what we have here are TTubules, transverse tubules, and they bring the excitement into the skeletal muscle cell. Each T-tubule is surrounded by 2 terminal cisternae. Terminal cisternae store boatloads of calcium, and they are studded with voltage-gated calcium ion channels, so that when the excitement, the muscle action potential, travels down the T-tubule, it causes the terminal cisternae to release the calcium, lots and lots of calcium. The terminal cisternae are part of the sarcoplasmic reticulum, which is the lacy sweater sleeve. They're part of the sarcoplasmic reticulum, but they're a specialized part because they have the voltage-gated calcium ion channels and even more calcium. The book says the SR has 40,000 times more calcium than the sarcoplasm surrounding it in a resting skeletal muscle cell. So when the excitement, a muscle action potential, travels through the cell, it goes around every T-tubule, which opens the voltage-gated calcium ion channels on every terminal cisternae. These 3 structures together, the T-tubule with the 2 terminal cisternae on either side, are called a triad. So muscle action potential causes the release of calcium. And there's a triad around every zone of overlap. So let's look at this beauty. So here we have a zigzag. A zigzag is a Z-line. Z-lines are made of actinin, A-C-T-I-N-I-N. Oh, look, it's another zigzag, which is a Z-line made of actinin. Attached to the Z-lines are thin filaments. We know thin filaments have a central rod, so this will be yellow here, of nebulin, and strings of pearls, 2 strings of pearls around each nebulin, coiled around like a double helix, and those are your G-actin, G for globular, like a globe, like spherical. And then on each G-actin, the part furthest from the nebulin, is a little active site. And the active sites are normally covered by a protein called tropomyosin, because it blocks the myosin. And the tropomyosin is held in place by these little green ones, and the little green ones are the troponin, T-R-O-P-O-N-I-N. And the troponin hold the tropomyosin in place until the T-tubule brings a muscle action potential, which opens the voltage-gated calcium ion channels in the terminal cisternae, which release the calcium. And then the calcium rushes out, and there's a triad around every zone of overlap. So the calcium rushes out and binds to the troponin. When the calcium binds to the troponin, it changes its shape. And each troponin has 3 little bits, one bound to the tropomyosin that covers the active sites on actin, and one that's bound to the nebulin. What's the nebulin? G-actin. Anyway, it holds it in place. So when the calcium binds to the troponin, it changes the shape of the troponin, pulling the tropomyosin off of the active sites on actin, which will be the little tips of these little beads. And the active sites are now exposed. Now these active sites on the ends of the G-actin are also called myosin binding sites, because what binds to them? The myosin. So here in the middle is the M-line, M for middle, and attached to the M-line are thick filaments. And the thick filaments are made of myosin. Myosin has a, tail, and then lots of little heads. So then the heads bind to the active sites on actin, and then they pivot on their hinge. They have a little hinge. So they pivot on their little hinge, and then, let me zoom out so you can see what I'm doing here. Zoom out. So they attach and pivot, and when they pivot, since they're attached to the thin filaments, they pull the Z lines closer together. So pivot, 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 pivot. And there you go, that's how the muscles contract. So let's talk about the parts of the sarcomere before we put this pretty model away. We've got Z line. Z line to Z line is a sarcomere. So this is one sarcomere. So that makes this one myofibril. In the center of the sarcomere is an area with thick filaments but no thin filaments. That's your H band or H zone. And then there's an area with thick and thin filaments overlapping. That's your zone of overlap. Here's another zone of overlap. So the length of the thick filaments is your A band, and where there are no thick filaments is your I band. So when sliding filament theory says that these thick filaments and thin filaments don't stretch and recoil like an elastic band, which is what they first thought was happening, that it's a sliding filament theory. The thick and thin filaments simply slide across each other. So let's keep track of our I band. Okay, I'm in the screen now. Of our I band, when the muscle contracts, the I band gets smaller. So now let's keep track of our H band. When the muscle contracts, the H band gets smaller. Okay, now let's watch the zone of overlap this time. When the muscle contracts, the zones of overlap get bigger. And what about the A band, which is the length of the thick filament? What does it do when the muscle contracts? Stays the same. The thick filaments each stay the same. The thin filaments each stay the same. So let's do a quick overview of how the muscle contracts. So we're going to start with this model with the axon. The axon is part of a nerve. The muscle only contracts when a nerve tells it to. Each muscle is controlled by one nerve at one neuromuscular junction, which will be the ugly orange splotch. And each axon ends at synaptic knobs, axon terminals, or synaptic terminals. And they contain these pearls called synaptic vesicles, and they are filled with a neurotransmitter called acetylcholine, abbreviated ACH, capital A, capital C, lowercase h. And then when the nerve action potential tells it to, it causes the exocytosis of the acetylcholine into the gap between the nerve cell and the muscle cell, which is the baby blue space. The gap is called the synaptic cleft. And then in the synaptic cleft is a chemical that binds an enzyme that breaks down the acetylcholine. It's called acetylcholinesterase, which is abbreviated ACHE. So just ACH, but throw a capital E on the end. And of the half of the acetylcholine released that reaches the, what's the wiggle squiggle? The motor end plate, which is a specialized area of sarcolemma, which is the smooth cell membrane. The ACH binds to ACH receptors, and that allows sodium to rush into the cell. Oh, here are mitochondria. I don't think we covered mitochondria on this model on the other video. Mitochondria are all the red hots. So then the sodium rushes in, and sodium always rushes in. Both the chemical gradient and the electrical gradient pull sodium in. And so the sodium rushing into the cell is a change in the transmembrane potential, which is the muscle action potential. So the muscle action potential sweeps down the entire sarcolemma of the cell, imagine a pool noodle, and also inside the cell on each T-tubule. And each T-tubule is touching two terminal cisternae, which are part of the sarcoplasmic reticulum, which store boatloads of calcium. And those three together are a triad. So The terminal cisternae have voltage-gated calcium ion channels and allows the stored calcium to rush into the sarcoplasm from the sarcoplasmic reticulum. And there's a triad around each zone of overlap, which is handy because it's the calcium binding to the troponin that allows the muscle contraction. And it does that because the calcium changes the shape of the troponin, which is bound to the G-actin and also the tropomyosin, and it pulls the tropomyosin off of the active sites on actin so that the little myosin heads can bind to them. And then the myosin heads pivot on their little hinge, pivot, and then since they're attached to the thin filaments, when they pivot, that brings the Z lines closer together and the sliding filaments slide across each other. So they attach, pivot, and then they have to bind an ATP to let go. And then they break the ATP down into an ADP and a phosphate, and that resets them. And then they reattach, pivot, drop off the ADP and phosphate, bind another ATP to let go, and they continue through the process, and that's how you flex your arm. Okay, so that's, I think, all of the muscle physiology. We will start back up next video with the muscles, the top of page 26. Yeah, so this has been A&P 1, Lab 3, Video 3. The education must go on. Dr. J out.